Hello, my name is Katherine Do and welcome to the video for Module 4 of the Clinical Metaproteomics tutorial. In this video, we will cover peptide quantification. The video preceding this one covered Module 3, peptide verification. If you are just joining us, please watch the previous videos where we provide background on the clinical metaproteomics field and delve into database generation, peptide discovery, and peptide verification. They will prepare you for this video. Now let's begin. First, we will recap the verification module and see where that puts us in the context of the clinical metaproteomics tutorial. Then, in the hands-on section, we will execute the quantification workflow. This is the shortest module as it consists of using MaxQuant for quantification and preparing the outputs to set up for the next and final module, data interpretation. Here is the overview of Module 3, verification. First, we took the microbial peptides we obtained in the discovery module and verified that they were indeed of microbial origin using PepQuery 2. Then, using Query Tabular, we queried these verified peptides for their protein accession numbers, extracted these accession numbers, also known as UniProt IDs, and used the UniProt tool to generate a database containing the verified protein sequences along with human and contaminant proteins. This verified database will be used in this video for quantification. Now let's see where we are in the tutorial. So we've completed 3 out of the 5 modules of this tutorial: database generation, discovery, and verification. We've now reached the quantification module. Performing quantification enables researchers to measure and compare the abundances of proteins and/or peptides in biological samples. It can allow researchers to decipher complex protein expression and protein regulation, which can provide insights into quantitative changes across different experimental conditions. Quantification also provides an additional quality control measure by validating initial protein identification and providing data normalization for increased accuracy. Here we will take the verified protein sequence database for Module 3 and use it as our input database for MaxQuant quantification. You may remember back in the discovery module, we used MaxQuant to identify peptides for verification. Now we will use MaxQuant to further quantify verified peptides, both microbial and human. We're not using Search GUI and Peptide Shaker as there is no capability to perform quantification of peptides and proteins. In this module, MaxQuant will give us various outputs, including quantified peptides and protein groups that are derived from human and microbial origins. We will prepare these outputs for the next and final module, data interpretation. Next is the hands-on section. Since I've already covered setup in previous videos, I will jump straight into starting the workflow, and while it is running, I will cover inputs, tools, and end on outputs. I've already prepared the history with the inputs and imported the workflow. Now we just need to select the inputs and click Run Workflow. Now let's cover inputs. From Module 3, we need the verified protein sequence database that consisted of protein sequences of our UniProt IDs that were merged and filtered with human and contaminant proteins. We will also need the sample MS datasets as raw files. If you remember, MaxQuant can process raw data. As I previously mentioned, this module is the shortest one in the tutorial. In summary, we will take the verified database and sample datasets and put them into MaxQuant to quantify verified peptides and proteins. Then we will select for distinct microbial peptides. Next, we will cover the quantification in more detail. Just as we did in Module 2, we have provided a file called Experimental Design for MaxQuant that will tell it how to organize and analyze our sample datasets. MaxQuant will search the peptides and proteins from our sample MS data against the verified database to perform quantification. The outputs we are interested in are the evidence file, protein groups, and peptides. The protein groups file will report all proteins that contain the sequences of identified peptides. Since our inputs contain a mix of human, microbial, and contaminant proteins, MaxQuant outputs will not differentiate between microbial and human sequences. For human proteins, peptides are shared within isoforms, so the protein group file reported groups of proteins that share peptides. For microbial proteins, protein groups are reported across organisms based on shared peptides. In order to perform data analysis of the microbial community in our sample data, we will have to extract microbial peptides from the peptide file. On the next slide, we will focus on the peptide file, but in the last module, we will also need the evidence and protein groups files to visualize and interpret our results. For the final step in this module, we will take the MaxQuant peptides file, select for microbial peptides, and then group to remove any duplicates. At the time of this video, we obtained 155 microbial peptides before and after grouping, so we did not have any duplicates. Please note that tool versions and protein databases are regularly updated, so these numbers may be different for you. Also remember that microbial peptides are in much lower abundance relative to human peptides in our samples, so we will get a much smaller number of quantified microbial peptides compared to the total number of peptides in the peptides file. In the next and final module, we will show you how to visualize and interpret the outputs from this module. This marks the end of Module 4, Quantification of the Clinical Metaproteomics tutorial. Please leave feedback on what we did well and how we can improve. To navigate to the next tutorial, go to the bottom of the GTN tutorial page for this module until you reach the Go Further box. Click on the link to go to the next module. We hope to see you in the last video where we will cover data interpretation.